So we'll give people maybe 30 seconds to join. While we're doing that, I will go ahead and just welcome everyone to, again, another installment of the Black and Cotton Bio series. I'll go ahead and turn the waiting room off here so we don't have to monitor that. But yes, this is the first seminar of 2021. So thank you all for still showing up and for your commitment to, supporting this great way for us to just talk about science. My name is Janae, and we also have Jane here who will be moderating the session. But just wanted to welcome everyone. And yeah, thank you again for joining. We have a great talk today with Dr. Regina Cordy. We also have some members present who read a paper of hers before today's meeting. So we're really looking forward to the discussion that is to follow. So yeah, just welcome everyone and take it away, Jane.
Hello everyone, my name is Jane Jerry. I'm from Kenya. I'm currently working on virus epidemiology at Emory Wellcome Trust. I'm here to welcome Dr. Regina Joice Cordy, who is an assistant professor in the Department of Biology at Wake Forest University. So Dr. Cordy received her Ph.D. from Harvard University in 2013 and conducted her postdoctoral research at Harvard School of Public Health and Emory University. So her PhD work in the group of Matthias Marti was focused on the study of Plasmodium Falciparum in human tissue using quantitative PCR and immunohistochemistry-based labeling approaches to localize parasites with respect to host cells and tissue microvasculature. So her postdoctoral work with Dr. Curtis and Dr. Mary was focused on computational analysis of large data sets, including the microbiome within a public health context and a metabolome of malaria-infected hosts. Dr. Cordy's current research focuses on investigating how fluctuations in blood metabolites and gut microbes affect malaria pathogenesis using a combination of computational, histological, and in vitro approaches. Dr. Cordy, go ahead with the talk.
Okay, thank you so much, Jane. I really appreciate the introduction, and just thank you to you and Janaea and everyone else who's been working what looks like very hard on this really exciting initiative of Black women in computational biology. So I'm just really thrilled to get the invitation to present here and to be a part of it. So yeah, so I prepared some slides, which are basically kind of based on the paper that I sent out. And I'm thrilled to have just heard that you all did a journal club on this paper and that folks have read it and maybe even have some questions. So I would just say I'll go through some slides, but feel free to just kind of jump in and let it be a sort of interactive discussion, because I'm definitely interested to hear what sort of questions and thoughts you have. And I'll also try not to take up the full hour so that we also can potentially have some time just for general discussion afterwards. I think Janaea mentioned that there might be some general kind of career type questions and things like that you all might want to also talk about. So hopefully we'll just leave a little bit of room for that at the end as well. So let me go ahead and get my slides up. Okay, can you all see that? Okay, great. All right, so yeah, so my name is Regina Joice Cordy, and I'll just be giving this talk today on really the theme of the work that I've been doing on metabolomics of acute and chronic malaria. And just to show you a little bit of images of kind of where I'm at. So I'm an assistant professor. I'm in my third year and I'm at the Department of Biology at Wake Forest University. In my position, I do a combination of research and teaching. So this is just some images of our lab and of the different parts of Wake Forest campus, which is split between sort of a more suburban area and an urban downtown area of Winston-Salem, North Carolina. and also showing one of our teaching labs where we work with students and really get up close and personal with the different parasites. So that's actually a parasitology class shown there. But today's talk really is going to be about sort of the research that I've been doing and that related to the paper that I sent out. And so this work is really kind of focused on Plasmodium Falciparum. So I just wanted to give a little bit of background, although I think many of you on the call have a lot of knowledge about Plasmodium Falciparum. And so this is just some kind of quick points. But essentially, there's multiple species of Plasmodium, and one of the ones that I've been most interested in during my research is Plasmodium falciparum. And that's because this is really associated with a large amount of mortality and morbidity. And a lot of this occurs in different parts of Africa and also Southeast Asia. And so one of the things that I've been curious about for a while and sort of at different phases of my career been interested in is how malaria infections can present in different ways. So you can see this sort of spectrum of disease is sort of one way to think about it. So you can get cases that are asymptomatic, that are uncomplicated, meaning there's fever and flu-like symptoms, and also more severe cases, which unfortunately are associated with vast morbidity and even mortality. And so one of the kind of central questions I have around various parts of my research are just trying to understand, in addition to sort of the immune-related and parasite-related factors that drive some of that variability, are there also other host factors that might be contributing to this range of disease severity? And so the project that I worked on in that second postdoc that Jane mentioned was based at Emory University. And this is kind of the springboard for where my research takes off from my independent lab, and also the paper that you all read is from that postdoctoral research that I did at Emory. So I wanted to give you some background on that project overall, just to kind of let you know a little bit about it, but also because I want to suggest to you that for those people who in the group might want to play with the data a lot, and since it's a computational biology group, I thought this might be of particular relevance here. So the project that we worked on was called the Malaria Host Pathogen Interaction Center. And it was a multi-year project funded by the National Institutes of Allergy and Infectious Disease. And the main goal was to try to generate these large data sets that could be applied to natural infections. So we had some individual samples from cross-sectional studies in humans, and then largely it's based on non-human primate studies. And so what we did is we would, over the course of these studies, and I'll show you more information about the study design there in a couple slides, but essentially we collected a large number of different sample types and then performed a variety of different multi-omic methods. So genomics, transcriptomics, metabolomics, in addition to clinical and flow cytometry related factors. And then we've put all of this data onto a public database. So if anyone is familiar with the website Plasmodium, for anyone who does work on malaria in the group, or if you work on other sorts of organisms, it's probably very similar to the DB website that you may use for your own research. But essentially within that Plasmodium, there's a link to what's called the MAPIC. So that's the abbreviation MAHPIC. And there you can download these data sets and you can work with them yourself. And so I would encourage anyone who is potentially interested in, if they have a certain part of, protein of interest or metabolite they're curious about, you may be able to look to see its trend in our data set, and they're all publicly available and open. And so I'm happy to, if anyone wants to contact me to help help them navigate those data sets. So let's talk more about the study of design and the paper itself. So this was work conducted primarily at Yerkes Primate Research Center, and that's in Atlanta, Georgia. It's part of the Emory University. And this particular institute is really specializes in research related to rhesus macaques. And so a lot of infectious disease, including some newer work on SARS-CoV-2 is happening at that site. They do a lot of different vaccine related work. And so for us, we're really interested in working with that model system because for malaria, the rhesus macaque infections do replicate a good amount of the sort of features of malaria infection in humans. So to kind of show you a little bit about what I mean there, and some of you would probably have seen malaria life cycle diagrams before, but I'll just kind of walk through it sort of quickly and just show you the overlap with humans and non-human primates. And so if you can see my mouse, essentially a malaria infection is initiated with the bite of an Anopheles mosquito, the injection of what's called the sporozoite stage, which is a stage that makes its way into the liver, where it undergoes a vast multiplication event. Then it eventually gets out from the liver into the bloodstream, and that's where it replicates in these 48-hour cycles and releases progeny into new red blood cells. So that's really where the disease part comes in, the fever, the anemia, et cetera. And then really to perpetuate the infection, at some point along the way, the parasite will transition from asexually replicating to sexually replicating. At that point, those stages, when picked up by the mosquito, can go on to fuse and undergo sexual development in the mosquito and prime that mosquito for another transmission event. So this is really requires biological processes in both the mammalian host and the Anopheles host. And what we see with the non-human primates with the rhesus macaques is that we do find a lot of similarity with the stages and sort of the general way that the disease looks. So there's sporozoid infections can be performed in the animals. There's there's the ability to look at replication events longitudinally. We do see sequestration of the parasites, and they undergo gametocyte conversion. And so there's a lot of overlaps and things that can be looked at there. And so as I mentioned, our group was doing this, gathering multiple different types of data to look at. And the one that I was specifically focused on was metabolomics. And so in that view, what I was really aiming to do is to look at the metabolic phenotype, or the sort of biochemical activity of a site being analyzed at a particular time. So that's sort of what metabolomics gives you. So if you are familiar with some of the other omics and maybe not metabolomics, then one way to think about it is if you're used to thinking about genomics, meaning a full collection of all the genes in that particular cell. Here we're talking about metabolomics and all of the different metabolites and their levels in a particular tissue. And in the case of this study, we were looking at plasma, so basically the bloodstream, and we wanted to look at the number and types of those metabolites present at different times during the infection. So essentially to look at biochemical activity at different times of infection. So just to kind of explain a bit of the complexity of this plasma metabolome. So when you've got a malaria infection, one way to think about this is that you've got the contributions of the host. So that's just the mammalian host, which obviously transcribes, and makes proteins and undergoes all kinds of metabolic pathway activities with its regular life and also with the immune response. So those will lead to a certain number of host-derived metabolites. Then there's various environmental factors that can also have large influences on the metabolome. And in the case of malaria infection, we're adding in another organism that is potentially multiplying in high levels that itself has nutrient requirements that it needs to take up from the plasma. It also lyses red blood cells and creates extra debris. And it has its own set of genes. So it has about 5,000 genes, and it can also produce its own enzymatic activity and pathways. And so what we are tracking really is this kind of complex host parasite profile over time during infection. And so some of the questions that we have, or some of the sort of areas we hope that this method may shed some light on, are trying to understand metabolites that are perturbed in Plasmodium infected individuals specifically. So if we compare malaria infection to non-malaria infection, are there certain metabolites we can identify? Also interested in how the metabolites differ across disease progression. So that's what you've read about in the paper, is trying to understand how they differ from acute to chronic infection. And in terms of how this kind of translates, or what are some of the next steps, you know, and a very sort of translational point, some aspect of biomarker development may be able to eventually come from a technique such as this. But also on a more kind of mechanistic level, we may actually also understand some things, some sort of factors that are contributing to malaria disease progression by just getting a better sense of what's actually happening in the plasma during those infections. So when we study parasites in the lab, we tend to keep them in these ideal culture conditions where they have all the nutrients they need. They have kind of sort of a perfect growth environment, but what we notice in these hosts is that that's very much not the case. It's a very dynamic environment. And so, you know, this also is a way for us to try to understand a little bit more about that environment. And so I'm going to focus on first the human study, the part of the study where we really focused on some of the human cases. And this was kind of what got us inspired to really dig into this question. So the study that we did this in collaboration with Jetsuman and Rapat from the Mahidol Vivax Research Unit in Thailand. And what we did is we had a study where there were a number of individuals with uncomplicated Plasmodium Falciparum, and we also had healthy individuals from the same area. But we added in this third group, which was this non-malarial febrile illness group. And the reason we did that is because we had this hypothesis that there might be a good number of non-specific changes that just happen in any acute febrile infection, and they may or may not be specific to malaria. And so that was sort of to help parse those out. So that's the study design. And then from these individuals, we had plasma samples that we collected and were able to study with a metabolome approach. And so there really, we were trying to get at what are some of those metabolic perturbations you know, that are associated with the Plasmodium falciparum infection. So what this looks like in terms of once we have the plasma sample, we're running these samples through liquid chromatography mass spectrometry. And the goal there is really to get this untargeted metabolomics data set. So just to talk a little bit more about what I mean by that, essentially what we're doing is we're taking the raw data from the mass spec run, and we're looking at this data in the form of mass to charge ratios, retention times, and intensity. And so this is a format of the data that has not yet been matched up with reference compounds. So it does not have annotations to it, but it does have chemical identifying characteristics. And that's what we're looking at here. And so we look at that large untargeted data set in order to identify signatures that appear to be differentially regulated, so up or down in some of those different cases that we're interested in. And so that's really where the heavy computational part comes in. So in terms of trying to say, okay, we've got this big data set, which does not have the annotations yet, and we want to be able to kind of winnow it down and identify possible biological pathways and activity. How do we get at that? And so the computational biology techniques involved include things such as MWAS, which is very similar to GWAS, but essentially using the metabolome instead of a genome. So looking for associations between a certain factor and metabolite signature. And then also doing different statistical analyses such as PLS, and also looking at PCAs and trying to study clustering effects to try to see if we see big patterns in the data. And so once we go through those, and I'll show you data on that, we then take these results and input them into a couple of different pathways. So one of those is called MummyChug, which is a pathway analysis software for this type of data. What that does is it helps us to identify metabolic pathways that appear to be enriched in that data set based on the untargeted data. And then the second thing we do is we look at putative annotations. And so that's also using reference databases such as such as, what is it called? Now I'm blanking on the name of it, but basically different databases that already have the compounds and their mass to charge ratios in them. And then we align it to see what that appears to be. And so once we get that, that's sort of our putative annotation data. We then go forward with the confirmation, and that's a more targeted approach. And so that's where once we've kind of winnowed it down to a few pathways of interest, we do look at reference compounds and really target and confirm a few of our results that way. And so this is a principal component analysis that is looking at this data that I just showed you 2 slides ago with that structure of the falciparum cases, the healthy individuals, and then that non-malarial febrile illness. And what we're showing here is this is a way of looking for clustering to see how different the individuals are from each other in terms of looking at their whole metabolome. And so in terms of the view of this, we're looking at how we're basically showing each individual icon is an individual's whole metabolome, and we want to look for clustering patterns. And so what we can see kind of right off the bat is that healthy individuals are clustered separately from both groups of sort of the illness categories generally. So the Plasmodium falciparum and the non-malarial febrile illness group, not 100%, but to a large extent are clustered separately. We also see that within those cases that are febrile, there is some separation between non-malarial febrile illness and the Plasmodium falciparum group. And so this kind of suggested to us that possibly there's some component that kind of separates out the healthy versus the acute ill group, but then further, there's also some component that also separates out the falciparum from this other group. And the other group is a diverse group in and of itself. So the non-malarial febrile illness group in this particular study is mostly viral diseases and also some undifferentiated febrile illness. And so within the context of that, we can't say a lot about bacterial illness in our particular study, but in the context of primarily virus related illnesses, we do see some difference. So then seeing that there is a difference, we start to get curious about, which metabolites are actually contributing to that clustering pattern. And so we looked at first to see what was in common. As I mentioned, our hypothesis was that there probably were metabolites that were kind of commonly perturbed in acute disease. And we did find that. So a couple examples. One was that kynurenine was enriched in both the non-malarial febrile illness and Plasmodium falciparum samples as compared to healthy individuals. And this, we believe, reflects the sort of a turning on of an immune suppression pathway, as kynurenine is a tryptophan metabolite, and this has been previously shown in a wide range of different diseases to be activated during acute infection. So we think we see definitely some markers there. And similarly, reduction in lyso-PC seen in a variety of different acute diseases, including sepsis, and has to do with the PLA2 pathway and also relates to sort of that vast immune response. And so we do believe that we are seeing some common signatures of acute infection that are seen in not just malaria, but other diseases. Then, trying to identify a few that do seem to be a little bit more malaria specific, and we're still working on this. This was sort of something that we're continuing to work on, and we have additional data sets. So we're still really pursuing this type of research. But in this particular study, we were able to identify biliverdin and sphinganine as those that seem to be more specific to Plasmodium falciparum than to the non-malarial febrile illness group of primarily viral illnesses in this particular study. And so what we believe we might be observing here is that biliverdin, which would be a downstream product of the release of free heme, which happens a lot during malaria. So malaria is associated with a lot of lysis of red blood cells. The body's going to be processing that and detoxifying it. And ultimately, we think that this signature of increased biliverdin is probably indicative of that hemolysis. And then with sphinganine, we're also believing that this is probably an immunological lipid signaling related event that we're observing here, and also may point to certain immunological signaling pathways that may be differentially expressed in viral versus, say, this parasitic disease. So just to kind of, that's just giving you a few examples just to kind of make it specific, but we did also do that pathway level analysis to kind of just generally identify what we see different between falciparum and non-malaria. And so there we see a large number of amino acid pathways. We do see hemoglobin related metabolites. and fatty acid related metabolites in that pathway level. So at this point, you know, and when we look at these sort of cross-sectional studies, you start to get to this point where it's very interesting to look at cross-sectional data in the sense that we're really identifying something that's happening in the patient that can be detected at possibly different levels in disease, and it kind of gives us that sense. But to try to understand a little bit more about the biology of what we're seeing, it's really more helpful to see if we can look at these metabolites in sort of a longitudinal way and associate them with different factors. And so that's why we ended up doing this study also in the animal models, where we were able to more carefully control the infection and look at the dynamics of these metabolites over the course of an infection. And so the goal there was to say, okay, we know that we see some metabolites that are definitely in a cross-sectional view appear to be at different levels in humans with malaria versus non. But what is the dynamic of these? And is it, you know, the entire phase of when they're infected or just partial phases? And are these changes associated with any clinical parameters? And so those are the kind of questions we wanted to explore using the animal model. And so here we're actually looking at a different form of Plasmodium. So just to kind of introduce that briefly, Plasmodium codnii is a different species of Plasmodium that infects rhesus macaques, but it is considered to be a good model for Plasmodium falciparum, which is really the main disease we're trying to study here. And the reason that it's a reasonable model, some of the reasons are it does exhibit a 48-hour asexual life cycle like Plasmodium falciparum. It's associated with the animals having an onset of fever and anemia in a similar way to the way in which falciparum malaria can have that onset. And there's also a possibility to look at acute and chronic phases of Plasmodium codnii infection in these animals. And so this is something that I think is very important in the context of Plasmodium falciparum, because in Plasmodium falciparum, there's also a large number of asymptomatic or mildly symptomatic cases in addition to the acute cases. And so similarly in this rhesus macaque model of Plasmodium codnii, we can look at essentially this asymptomatic type disease as well as the acute disease. Okay, so I'm going to show a few of these diagrams. So I'm going to kind of slow this down for just a second and walk you through one of them. So just to kind of talk a little bit about the study design. So the rhesus macaques, the way that we do these studies is that they're infected and they're studied for 100 days post-inoculation. So if you think back to that life cycle that I showed, they're inoculated with a sporozoite. And so they have this pre-patent period, which is basically where the parasite is actually in the liver stage. So the parasite is undetectable for about 10 days. So the black dots here are parasitemia, the red dots are hemoglobin. And so those are the factors that we can measure with a blood smear. And so that's what you're seeing across this longitudinal time course. And so what happens is once those parasites emerge from the liver, they rapidly increase and they reach what we were calling peak parasitemia. And that's the point at which we were giving them a subcurative dose of artemether. And the purpose of this was to knock down parasitemia so that the animals didn't progress to a more severe form of the disease, but it didn't eliminate parasitemia. And this is important because this is essentially what allows us to model chronicity. So the parasites are knocked down to sort of this medium level, and then they persist into this chronic phase. And then what we did is we defined these phases based on different clinical parameters. So the pre-patent phase is before they're out of the liver. The acute phase is basically the progression up to that subcurative treatment. This sort of post-subcurative treatment phase is everything where we still see parasites kind of going up and down above 1,000 microliters per 1,000 parasites per microliter. And then what we did is based on some literature that suggests that asymptomatic infections in humans persist at around 1,000 parasites per microliter. We just use that as a way to designate, okay, at the point at which parasites are maintained at that level or lower, we consider that chronic phase. And we also noted that that phase, as defined by parasitemia, was also associated with an increase in hemoglobin. And so this is kind of where you start to get into that kind of model of, sort of chronic malaria in these animals. And so we did this study in 4 different animals and 4 different rhesus macaques. These are outbred animals, and so they do have some variability within them. They do have the same, they were inoculated with the same strain and same inoculum of Plasmodium cotonii, but we do see variability in their clinical phenotype, probably due to their genetic backgrounds being somewhat different. And so that was sort of why we had to define these define these phases based on clinical phenotypes, because the animals themselves differ between each other. So this kind of shows the range of what we saw. We saw everything from an animal that sort of self-controlled parasitemia fairly quickly and maintains this low chronic phase for a very long time, and all the way up to individuals sort of in the middle, to an individual that required multiple subcurative doses in order to finally control. And so you do see definitely a range of severity. And that in and of itself is kind of interesting to us, because these are all malaria naive animals, similar age, like I said, similar, same parasite and inoculum, but just different host backgrounds. And so that also kind of intrigued us as to what were some of the underlying host differences that might control, might sort of drive an individual towards a more severe or more mild infection. And so then what I became very interested in doing was saying, well, can we look at the metabolome during these infections in that longitudinal way? So since we have these longitudinal infections, we can look at malaria, we can look at the baseline of the same animal and all the way out through the acute and the chronic phase and see what the metabolome looks like. And so there, what we did is we used the exact same methods. So the kind of untargeted metabolomics methodology in order to study the untargeted metabolome across these longitudinal time points. And so just like I had shown before, this is a principal components analysis showing these in each icon representing a distinct animal and time point, and then where they fall is kind of how similar they are to each other. So we're looking for clustering patterns in this diagram. And so again, kind of like what we saw with the human study, the uninfected time point, which is the baseline, we see that of all 4 animals grouping together to the right side of the graph. And then what was kind of, we saw next was actually a little bit surprising to me. I had hypothesized that the acute would be sort of all the way by itself, but what we actually saw is that the acute and the post-subcurative treatment were overlapping in this analysis. And what actually was quite different was the chronic phase. And so what this sort of suggested was that the point at which parasites were kept in this very low level seemed to be also linked to, associated with this difference in overall metabolites in the bloodstream. So just showing that a different way, this is a heat map in which you can see to the left, this is grouping the same colors that are used here. So these are the baselines for all 4 macaques. This is the combination group of that acute and post-treatment group. So that's the data is what really showed us that those were actually fairly in common from a metabolome view. But then what we did see in terms of a shift was the chronic phase. And so that really kind of suggested that there may be a difference in the chronic versus the more acute-like metabolome phenotype in malaria. And so we then, as I mentioned, since this is a longitudinal study, one thing we could do that's pretty intriguing is that we can really track these metabolites over time and kind of look at their pattern. And so here I'm just showing one of the metabolites that I talked about at the beginning, one of the lysophosfatidylcholine species. And you can see that it sort of drops in all 4 animals, it actually drops somewhere in that acute to post-subcurative time period. And in all animals, it recovers back during the chronic phase. So I thought that was pretty interesting. That's pretty striking in terms of how different it is during the acute versus the chronic phase. And also to some extent, there even seems to be some variability between those animals that appear to control more quickly than those that controlled more slowly. Now, we only have 4 animals here. We can't say that statistically, but there may be some trends there. We also see the other side of that, which is certain metabolites are increased during acute infection and are lower in chronic. So that sphinganine metabolite that we also saw increase in the or higher in the cross-sectional study, we also see that in 3 of the 4 animals, it goes from a lower to a higher amount in the time point 2, and then it comes back down to baseline after that. So it really is not all phases of malaria is this metabolite up. It's really kind of a temporal pattern. So there we also looked at the pathway view just to kind of give us this sense for whether we were seeing similar pathways in the rhesus macaques as in the humans. And we did see a number of the same types of metabolic pathways perturbed. So amino acids, hemoglobin-related metabolites, fatty acids also showed up in that analysis. And so that kind of motivated us to look into the reference compounds that we wanted to use to sort of validate some of these findings so that we could see whether there were any particular markers that we could sort of pinpoint that distinguished acute and chronic disease. And so that's where we kind of in that last step, we went ahead and we did this approach where we looked at a number of different metabolites that were spanning the metabolic pathways that were showing up in our pathway analysis so that we would kind of, use that untargeted approach as a way to sort of narrow down what to look at in the reference compound based approach. And then we took a number of the samples from the rhesus macaque and from the Thai human study to run in this approach in order to kind of see what those looked like. And so just to show you a little bit of that data, and that was it, we were able to really quantify these metabolites more specifically here with the reference compounds and standard curves. And so here what you can see is some amino acids such as arginine, you see a decline at the acute phase in rhesus macaques, and similarly you see the lower level in humans. Then looking at that, the lipid lysophosfatidylcholine, you similarly see sort of a lowered level in the rhesus macaques. Here it actually looks like you do see a recovering, whereas in the human side it's a little bit less clear. And then we also see this increase in certain carnitines during the acute infection and sort of a general higher level in the acute infection in humans. And other lipids such as sphingomyelin, we also saw what looked like a depletion kind of setting in during the chronic phase. So that was also pretty interesting, something that wasn't significant in the baseline to acute or the healthy to acute, but was significant in chronic. So that might even represent something related to how is chronicity maintained specifically if it's not as affected during acute. So just to kind of summarize that, when you look across the human and the rhesus, and we kind of tried to look at them both basically because both have sort of upsides and downsides. So in the case of the human study, the good thing is that you're looking at real Plasmodium falciparum, but it's a cross-sectional study of humans. They have all different factors, diets, different ages, activities, all kinds of things. So those are sort of caveats. On the rhesus macaque side, we're obviously looking at a model organism, but we do have a much more controlled longitudinal setup. And so our thought was that by looking at the intersection, we could kind of have ideally some more robust signatures. And so kind of overall what we see is a couple of amino acids that do stand up to that overlap, glutamine, arginine, multiple lysophosfatidylcholines that are depleted, and also this enrichment of acylcarnitine, and then multiple lipids are also depleted in that chronic phase. So just to kind of summarize, and then I'll just kind of briefly touch on next steps. So some of the things that our lab is doing to follow up on this. So just generally what I've talked about is this cross-sectional study. We were able to see malaria infection exhibiting definitely some common features with other febrile illnesses, including kynurenine and glycophospholipid changes. We also saw some specific in our study. So in comparison to the mostly viral group, we did see some signatures that looked to be more specific to malaria. So sphingolipids and hemoglobin-related metabolites. We also see that in general, the amino acids, fatty acids, and hemoglobin-related metabolism is driving a lot of that signature. Then looking at this longitudinal study in rhesus macaques, we could see that the metabolome did vary by clinical phases. So that was somewhat interesting that we could actually look at in a longitudinal view. We did see some overlap in the pathways between rhesus macaques and humans, and in particular, glutamine, arginine, multiple phospholipids, and acylcarnitine do seem to have this differential levels in acute versus chronic. So some of what we're doing next is we're trying to kind of broaden this study as a way of really trying to get even a better idea of these plasma metabolite changes and how robust they are in diverse malaria species stages and clinical states. And so we're working with a number of collaborators to obtain additional untargeted metabolomics data from cross-sectional studies of humans. The goal is to have data from multiple countries and multiple species of malaria as well, and clinical phenotypes. And the idea there would be ideally to have sort of a better gauge in general for markers that really are specific to malaria, and also maybe are even informative about the stage or length of time of infection. And in terms of the computational side of that, what's really important, as I'm sure a lot of you know, is to have a validation in an independent sample set. And so that's something that we're working on. And then the other thing, so that first one is very much a computational based project, but the second one here is also what we're doing in our wet lab, which is that we're following up on some of those results and trying to understand a little bit more about some of those metabolites that are perturbed, such as the amino acids like glutamine, and how those are affecting the system. So really looking at some of those perturbations in vitro in a Plasmodium Falciparum study, and also looking at how those are affecting the red blood cell. So I just wanted to kind of insert this little plug here that you know, my lab is really has a focus on graduate student driven research. And so I do have, I'm affiliated with the biology graduate program, as well as the microbiology and immunology graduate program. So for anyone who is interested in potentially applying for the program, please let me know. But we do have projects within this on those kind of type of topics that I just mentioned as graduate student projects. So I'll just kind of finish with some acknowledgements. So a lot of the work I talked about was at the Yerkes Primate Research Center with Mary Galinski, who's the PI of that overall study. Alberto Moreno is a co-PI of that study. Jetsuman and Rapat from our Mahidol Vivax research unit collaboration, and Dean Jones and Karan Upal, who are at the Emory Clinical Biomarkers Lab, and they're really the ones that have set up that pathway, the pipeline for analyzing untargeted data. And then going forward, my lab at Wake Forest University, it has right now has undergraduates and graduate students that are working on different aspects of trying to move this work forward. And just want to also acknowledge the overall NIH contract that funded the MAPIC and the NIHK award that's helping me to continue doing this work. So yeah, with that, I think I'll go ahead and stop and see if you all have questions. And thanks for your attention.
Thank you very much, Regina. That's very nice, nice talk. And if anyone has any question, please feel free to post in the chat or ask directly.
And feel free to ask about anything also. I think, yeah, someone had mentioned something about career questions. So whatever is helpful to you all.
Hi, this is Amanda. I'm sorry. I'm learning about this and it's really incredible. I knew nothing about malaria until I read the paper. And so one thing that I wanted to ask is pertaining to metabolomic biomarkers. So unlike the conventional clinical analytes, these profiles, they affect like they are influenced by a bunch of demographic, environmental factors. that can affect many various like values, and they increase the risk of false biomarker discovery. So I wanted to know, in your pipeline or while you're setting up your test, is there a specific parameter or parameter that you guys need to that you implement to decrease the false biomarker discovery? And how do you guys go around that issue?
Yeah, I think that's a wonderful question. It's you know, it's definitely the key important question in terms of trying to make sure you have a good biomarker. And so I think that one thing we've already started to realize that makes this really challenging is, like you said, the variability between humans. So diet and particularly the gut microbiota are a huge factor here. And one of the things that makes it complicated is that, let's say we get pretty good at trying to parse out something that's more likely human derived versus more likely parasite. Well, there's often still times overlaps between parasite and some different microbe of the gut microbiota. I mean, there's even eukaryotic commensal microbes. And so the thing that I think will make it challenging is actually the idea of a truly Plasmodium specific one is still challenging. And it might even be that what the strength of metabolomics might do is it might be the protein-based approaches that are being used right now for biomarkers for Plasmodium, you know, maybe with some adjustments may still be the way to go for the Plasmodium part. But then what I think we are actually, I would say the metabolome side seems to be a little bit more, if there is a real sense for it, would be sort of adding in this second biomarker that might be indicative of this kind of phase of infection or progression of infection or something else like that. And so I think, to me, where I see the strength would be when you think about adjunctive therapies. So there's a number of investigations on the possibility for adjunctive therapies for severe malaria. And so let's say if we see you know, any of those really you know, needing to sort of classify patients a little bit better to target those adjunctive therapies, I think, you know, we may be able to come up with sort of a smaller boiled down set of metabolites that, you know, might on their own be able to kind of help classify that. So, you know, I would say many levels back from that in terms of just trying to see what the data shows us. But when it comes to, you know, actualizing it into an assay, there's a lot of challenging questions there. And especially, I think, when it comes to the gut microbiota and just how much variability there is, I would say it's even more confusing and in terms of finding specific metabolites.
Oh, that is really interesting. May I ask one last question? And I promise I'll be quiet. Send you an email. This is really incredible stuff. I just want to ask one more question quickly, because I saw the variability of the various biomarkers, and then you have acute biomarkers. So you classify them in different categories. And I wanted to know, these ones that you are targeting especially, where do they fall under the NIH's best biomarker classifications? Are they prognosis, diagnostic, or what type of biomarkers are these? I mean, there's so much potential for a database for these type of biomarkers. So that's why.
Yeah, no, I think, you know, it's interesting. So I've been kind of thinking in my mind, and this is, you know, I mean, subject to many levels of, you know, it would have to be really validated, but there are, I think I would expect there's more likely to be a prognostic application for it. Because I think that that's what in working with some of the clinicians, you know, it seems to be, if there is a need for additional kind of, you know, typing, it would be something like trying to see, you know, where is this person in that disease progression? Are they kind of pre-peak or post-peak, or, you know, like where are they? And so I think there's always the biological side, and that's another aspect of just doing these studies, is to try to just understand the biology. But I think the idea of sort of this personalized medicine approach of saying, you know, yes, we have an anti-parasitic drug, but you know, there's also a wide amount of host pathways that are really kind of driving some of the actual disease itself. And so, you know, if we start having a need to sort of better classify those individuals, And even in a sense of just thinking about it as a resource, just do a better job of even allocating your resource. It might be that a little extra prognostic, could sort of aid in figuring out, oh, this might be that group that could benefit from, a particular adjunctive therapy. So I think that's very much like a, I would say we're not very close to that today, but I think that's the kind of direction that it could go in. The other thing I would say is that I've started to network a little bit with people who are doing similar studies in things like sepsis, and also are really interested in trying to come up with a better way to just look at non-malarial febrile infection. So basically just to say, here's a kid, they're febrile, we have no idea where to start. That's kind of one of the big applications that I would say across fields has become clear. And we all are seeing fairly similar signatures with that whole thing I was talking about. So like in the sepsis field, they're seeing similar, arginine and lysophosphatidylcholine. So we can definitely, in a way, I think at some point kind of combine into saying, this is just what illness looks like, but then probably beyond that, see if there's a way to differentiate some of them. And that, of course, is another thing related to, resource allocation and prognosis, is saying, is this person, what sort of prediction do we have of what's causing disease, and what path to take. And I think the reason, and this is what I've learned from talking to the clinicians about the reason why they believe this is important, is because in areas with high malaria endemicity, detecting the parasite might not mean a lot. It might mean, well, a lot of people might have the parasite, but they might be asymptomatic. So if they really are sick because of a bacterial infection, then you really want to know that, And so just finding the parasite could actually be something that could be harmful, because it might make them think that the parasite's causing disease, and then an anti-malarial might not help. So I think that's where that field is also kind of going as a broader goal, is to say, if just febrile infections can have kind of this like multi-platform to just help direct it to, a parasitic or a bacteria or a viral path, that would just kind of help, better treat individuals. And so, yeah, that's something that I would say very recently we've kind of been in some conversations about, and trying to figure out how we can, think about these things.
Thank you for your answer.
Sure. Oh, is there something in a chat? Let me see. I can't see it. It looked like there was a chat, but I can't actually see the window for some reason. If there is one, if someone could read it.
I can help you out. So it's a question from Nasha. She's asking for the result where you saw clustering of the post-treatment and acute phase samples. Could that be explained by therapeutic efficacy or some type treatment resistant phenotype?
Yeah, so the issue with the treatment in the animals is definitely something that can be, is some level of a confounder in the data. I think the thing that actually is interesting to us is that even though I was actually hypothesizing that we would see the biggest difference going from the acute into the post-treatment, what we actually saw was the biggest difference in metabolome in the acute versus the chronic. And so then if you look at the number of days that has passed since the subcurative treatment, we're now out of the window of where we would expect that drug to be in pretty high levels. So I guess the way we've been thinking about it is that yes, there's definitely an effect. And in fact, you can see it on the parasite. So this paper also has parasite transcriptional data, and you can definitely see that it has an effect on the parasite. But we also see in chronic this kind of both on the parasite transcript side and on the host metabolome side, shift that seems to be independent of the drug. And so, yes, I do think there's definitely an effect, but I believe that it doesn't appear to be due to, it doesn't appear to be driving what's happening in the chronic phase. That's our impression, but it's a really good question. and one of those things, unfortunately, we couldn't do a lot about. It's definitely, you know, falls in the category of in a model system trying to find a good way to, you know, longitudinally look at these sort of two phases of infection and sort of force it to occur. You know, obviously has some caveats, but I think we tried to look at that in different ways to sort of parse that out. Any other
Well, if there are no more questions, are there any?
Hi, Beth. Just saying hi. Nice to see you.
Yasha has one more question. More broadly, what are some challenges with doing primate-related research? Are there N equals 1 to 5 studies typical in this field?
Oh yeah, well, I think you nailed the one of the big problems right there is that typically they're small number of animals, and I would say, is it typical? I mean, there are some studies that definitely have more animals, but I would say that was the real critical reason why we had to do this as a longitudinal study also in them, is really to be able to sort of compare them back to themselves. But obviously any comparison between the animals, I mean, we really can't, you know, we have no statistical ability to do. So yeah, I would say there's a challenge in terms of the cost, feasibility, you know, in terms of trying to do large scale work. But some people do manage to do it. But yeah, I would say that's definitely a challenge. And I think that it's also just, you know, it's probably, it's not a necessarily something easy for a lab to also just kind of set up, say, on their own. So like the fact, that's why I mentioned the Yerkes Primate Research Center, is it's really a center that specializes in working with rhesus macaques. They have their own colony of rhesus macaques and a very large number of staff that are veterinarians and veterinary technicians and are really dedicated to that work. And so it's something that I don't maintain in my own lab in the biology department at Wake Forest, for example. It's to me, that's just too large of an undertaking. But so now actually what I do in terms of kind of moving forward with that is that the online data set that I mentioned at the beginning, the Plasmodium, I'm kind of in the process of, because we generated so much data and no one can possibly look at all of it and at once, I'm sort of treating that as kind of this virtual way of studying the, you know, animal signatures and kind of analyzing it, but it's essentially locked in time. So it's not repeating new experiments when I have a new question. It's basically reinterrogating an existing data set on non-human primates. So I would say it's very challenging, and I think you have to be really think through, you know, the resources and how you can do things, and are there any ways that you can, find a way to answer questions with small amounts of animals, or reuse data sets, or, you know, and if we have any tissue samples, you know, we archive all of them and save them. I mean, you really have to think about it very carefully. So I would say it's definitely a large undertaking, and in my opinion, best done with, you know, centers that have, you know, long-standing expertise in that.
Any other questions or comments? Well, thank you so much again, Dr. Cordy, and thanks everyone for being here. Yes, we have one more seminar for this month. You can go ahead and register on our website. We have 2 more in February. We have a pretty packed schedule. So as long as you're on our mailing list, you'll get updates for those as they come. But thank you everyone for sharing this space. We're really looking forward to continued journal clubs and continued community building through talking about science. So thanks again, and hope everyone has a great day.
Thank you so much for the invitation, and congrats again on setting up such a wonderful group and program. This is amazing. I'm really happy to take part in it. So be in touch, anybody, especially if you're looking at grad programs, or if you think of questions later. I'm always available by email. Thank you.
Thanks, everyone. Bye.
Bye.